All right, we can get started now. Thanks everyone for joining. Welcome to SILCS Bio's 2024 software release webinar. I'm Nathan, sales manager at SILCS Bio, and we're excited to introduce version 2024, our latest version of SILCS. So to give some history, SILCS Bio started in 2012 with a few research papers from the McCrell Lab at the University of Maryland, Baltimore. We've since grown, adding new features every year and growing our capabilities to service big and small pharma with service projects, as well as provide software products to improve computational drug design process for them. Our efforts have increased the accuracy of computer-aided drug design while still being computationally efficient, which is the core mission of SILCS Bio. Through user feedback from you all and new research from our co-founders, we've been able to improve year over year. And today's webinar demonstrates that feedback and new research that we have incorporated in our software. So the agenda for today is, we'll first introduce our team, then we'll go over what SILCS is and the new 2024 features, and then we'll have a live Q&A with our team using your questions, which you can submit in the Q&A box below anytime during the webinar. So firstly, I'll start with introducing our team. So we have our three technical team members, Oscar, Abhishek, and Oliver. They're our computational chemists at SILCS Bio. And then we also have our two co-founders, Dr. Alex McCrell and Dr. Olgan Guvench, who are also on the call as well. And they'll be here to answer those questions during the Q&A portion. So without further ado, let's get into the webinar presentation.
Can everybody see this screen?
Hello everyone, welcome again to the SILCS version 2024 release webinar. Let's get started. Today we will be going over these topics. For those who are new to SILCS, we will briefly introduce the SILCS method. Then we will share a validated workflow for using our small molecule suite in your drug discovery pipeline. We will then take you through some exciting new features we are releasing in version 2024. Lastly, we will talk about what's in the development stage and end the webinar with a Q&A session. Okay, let's begin with an introduction to SILCS. The fundamental idea behind SILCS, which stands for Site Identification by Ligand Competitive Saturation, is to use simple chemical solutes to map the functional group affinity of the entire protein. The probe molecules, along with water molecules, compete with each other to occupy the most favorable interaction sites on the protein surface. We extract this information, analyze, and process it to generate a pre-computed ensemble of affinity maps, which we call frag maps. We will show how this one pre-computed ensemble can then be used for various tasks in drug design campaign, such as site identification, virtual screening, hit identification, hit to lead, and lead optimization. Here is what undergoes in a SILCS simulation. We start with a protein and immerse it in a box with SILCS probes. Note that there is no ligand present in this case. 10 systems of the solvated target protein with scrambled side chains are generated as starting structures for 10 independent runs. We then run each of the 10 systems for 100 nanoseconds of MD simulation. After 1 nanosecond of MD simulation, we introduce an enhanced Grand Canonical Monte Carlo, that is GCMC simulation, for 100K steps before resuming the next MD simulation. Here, the MD simulations allow for conformational sampling of the protein as well as the probes and waters. And the GCMC simulation allows for sampling of probes in the deep pockets. The information from this huge calculation is then processed to generate the frag maps as shown on the right hand side here. To do that, we divide the full 3D space into tiny cubic voxels and calculate the probability of each probe occupying the voxel. Now here is a representation of what we find in the presence of a protein. The probability of the probe molecule occupying a given voxel is driven by its favorable and unfavorable interactions with the protein. There are regions with higher and lower probabilities than the probability of probes interacting with each other in the absence of the protein. And importantly, there are regions with no probability. All these probabilities are then normalized with the bulk solution probabilities and Boltzmann transformed to get a grid free energy at each cubic voxel. This is done for all types of atoms in the set of probe molecules, giving us the final frag maps. Now, we can visualize these frag maps in reference to our starting structure, visualizing different frag maps that represent apolar, hydrogen bond donor, hydrogen bond acceptor. Positively and negatively charged regions can be quickly analyzed for qualitative assessment of your target. Additionally, different GFE levels allow for regions that will bind the specific chemical class most tightly to be identified. Unfavorable regions may also be identified. Now, to use frag maps in a quantitative way, we first take the ligand pose and overlay in the field of frag maps. We then take each ligand atom and assign a frag map type to it. Then, based on the location of the atom and the grid free energy at that location, we can then get an atomic contribution to the ligand's energetic favorability. Basically, we can get an atom-based free energy contribution that can be summed into a score that we call LGFE, ligand grid free energy. This LGFE score is an estimate of binding free energy of the ligand in that pose. Because the frag maps are pre-computed, this simple assignment and summation is instantaneous and becomes the core of this technology as we move forward. The SILCS technology is not limited to overlaying ligands onto frag maps. We can also subject the ligand to the conformational sampling using what we call SILCS Monte Carlo or SILCS MC docking. In MC docking, we sample the translational, rotational, and torsional degrees of freedom of the ligand in the field of frag maps. The Metropolis criteria uses intramolecular energy of the ligand conformation and the LGFE to optimize the function of finding the best ligand pose and most favorable LGFE score. We can do this in a full docking mode where we are sampling the full rotational and translational space of the ligand in, say, a binding pocket, or in a mode we call pose refinement, which is more local relaxation from a known starting conformation, such as from a core crystal structure. The full docking takes typically an order of approximately 1 minute or so, representing a speed up of over 1000-fold compared to traditional free energy calculations. Here are results from a recent validation study that compares the free energy perturbation methodology with SILCS method. We have presented a comprehensive study of approximately 200 ligands across 8 different targets, where we compared SILCS with number of previously published FEP studies. It is very evident that LGFE scores provide highly comparable relative binding affinity to the free energy perturbation calculated delta G values. And when using iteratively with experimental data, and this experimental data can be for small number of compounds as small as 10 molecules, we were able to improve the relative ranking of the ligand affinities shown by the percent correct data here on the right column, where for standard SILCS the percent correct was 0.71. With SILCS MC machine learning optimized approach, we can get it up to 0.81, which is on par and above the standard free energy perturbation methods that are out there. To summarize the key advantages of SILCS bio approach, it is the pre-computed GFE frag maps, allowing rapid and accurate ligand binding affinity estimates in a way that leads drug design process. And beyond the quantitative predictions, usually the frag maps easily allow for an understanding of types of functional groups that can be added to a molecule to increase its binding affinity, which is very useful in the medicinal chemistry to give ideas about synthetically accessible molecules. We note that frag maps explicitly count for protein flexibility. This is important because we know the protein can undergo local conformational changes in presence of different ligands. The frag maps account for desolvation effects, and this refers to the normalization procedure which we described earlier. In addition to globular proteins, we also support membrane proteins such as GPCRs, ion channels, and transporters. We also have a specifically optimized protocol to apply the same method to RNA targets.
Now that we have the basic understanding of SILCS technology, let's take a quick look at the architecture of the SILCS Bio software suites that meet your needs. SILCS Bio offers 4 different suites. The small molecule suite applies the novel SILCS methodology to cover the conventional drug discovery process and includes SILCS frag maps, SILCS hotspots, SILCS farm, SILCS MC docking plus ligand optimization, and the SILCS BML programs. The biologics suite combines SILCS frag maps, SILCS hotspots, and the SILCS PPI in a single tool to build models for biologics formulations, which aim to reduce the protein-protein interaction and aggregation, and improving viscosity and stability, among others. The SSFEP suite applies single-step free energy perturbation to deliver free energy calculations orders of magnitude faster than conventional FEP. This enables the evaluation of large numbers of modifications to a parent ligand while maintaining accuracy. The CGenFF suite contains the well-known CGenFF program to rapidly assign charm general force field topologies and the parameters for novel compounds. The CGenFF suite also contains CGenFF covalent for compounds covalently bound to proteins and a module for conventional MD simulations. All of these suites have been integrated into SILCS Bio graphical user interface. In the small molecule suite, like the traditional computer-aided drug design process, an input structure is required. This structure can be experimentally determined from crystallography, NMR, or cryo-EM. You can also use homogeneous modeling or AlphaFold to predict the initial structure. This initial starting structure is used as input to SILCS simulations which generate the SILCS frag maps. These frag maps are the basis for the rest of the small molecule suite. With SILCS hotspots, you can identify any allosteric binding site or adjacent site if you know where the ligand binds. If you don't know where the binding site is, you can choose the top-ranked binding site to start. With SILCS farm, you can convert the SILCS frag maps at the given site into truly target-based pharmacophore features and virtually screen a prepared 3D compound library based on the features. With a known or putative binding site, you can use SILCS MC to perform ligand docking and post-refinement. After the experimental testing with SILCS BML, you can build Bayesian machine learning models to refine the SILCS frag maps weights based on the experimental data for further ligand optimization. To set up the SILCS simulations, you can choose the SILCS Bio GUI to easily click through the process with a connected HPC server by SSH protocol, or you can use a command line interface with more advanced options on the server. Both GUI and the command line interface support multiple target types, from globular proteins, membrane proteins, to RNAs. Once the SILCS simulations have been completed, you can visualize the SILCS frag maps using SILCS Bio GUI along with the target structure in different representations. These frag maps are useful not only for qualitative analysis, but also for quantitative analysis. Here is an apo structure of the tRNA methyltransferase and its corresponding SILCS frag maps. Overlaying the structure of a known ligand shows that the SILCS simulation successfully generated the hydrogen acceptor map around the oxygen atom and the hydrogen donor map around the nitrogen atom without the presence of the ligand. We can use the atomic GFE values to see the contributions of each atom of the ligand and inform further ligand optimization. Adding a benzene group to fill in the apolar map shows that the GFE of each carbon atom contributes to improve the binding affinity. SILCS hotspots can identify the binding pockets, including allosteric binding sites. In SILCS hotspots, a fragment database is docked in and around the protein to identify the potential binding sites. You can use your in-house fragment databases to detect the binding sites that have more desired features, and you can also use the FDA-approved compounds to validate the drug-like binding site. The SILCS BioGUI provides a convenient way to perform SILCS hotspots and visualize the results. SILCS Farm can accurately generate the target-based pharmacophore features based on the SILCS frag maps at the chosen binding site. The pharmacophore generation is ultra-fast, which only takes a few seconds on a personal computer. You can manually select and deselect the features and adjust the radius for certain features as needed. The exclusion map, shown as small beads, can also be included for a pharmacophore-based virtual screening. The pharmacophore feature-based virtual screening included in the SILCS Bio platform can easily handle millions to billions of compounds. Finally, in SILCS MC, you can perform ligand pose refinement, exhaustive docking, or ligand optimization. All docked poses or ligand modifications will be ranked by LGFE, and the atomic GFE contributions of each ligand will be available for visualization. With the subtle atomistic GFE contributions in your hands, you can either do a quick pose refinement to generate a more fit pose, or further refine the ligand using SILCS MC ligand optimization. The accuracy of SILCS MC is at the same level as traditional FEP methodology, but only in a fraction of the computing time. The SILCS BioGUI enables you to perform SILCS MC ligand optimization with just a couple clicks. All you need to do is click on the add-on to modify, choose R groups you wish to apply, and run the pose refinement for the newly generated ligands with delta LGFE values for all poses. After the runs are complete, modifications leading to favorable and unfavorable delta LGFE will be represented in orange and blue bars respectively. And you will be able to click on each modification and the add-ons of interest to see the atomic GFEs and get a quick summation from the ligand analysis panel.
Next, we will share a case study that successfully used the structure-based drug design workflow in SILCS small molecule suite. In this recent publication published in PNAS, the researchers applied SILCS to study the beta-2 adrenergic receptor. Here the researchers started with a novel conformation of beta-2 AR, an intermediate between active and inactive states. They generated the SILCS frag maps for this protein conformation. The frag maps were then used to generate hotspots by using SILCS hotspots, where nearly 100 mono and bicyclic chemical fragments are docked comprehensively in the field of frag maps to generate the frag map binding poses that encompass the entire protein. These poses are then processed with 2 clustering rounds to identify these hotspots. These hotspots were then analyzed and used as the basis of defining the putative allosteric site. This process of defining the putative site can draw support from any experimental data available among recommended practices of looking for collections of adjacent hotspots in which fragments occupying those sites could potentially be linked to create drug-like molecules. Once the binding site of interest is decided, using the SILCS farm module, they generated target-based pharmacophore features from frag maps in that binding site. These features were then grouped into 8 different pharmacophore hypotheses. 2 drug-like databases were subjected to pharmacophore screening using these hypotheses. Top 10,000 hits per hypothesis, that is 80,000 total, were then subjected to Monte Carlo pose refinement to get the refined poses and LGFE scores. Ranked by the most favorable LGFE scores, top 1,000 compounds were then selected for further filtering for paints and clustering to remove highly similar compounds. Remaining compounds were then processed to get a 4-dimensional bioavailability score and ranked again. Lastly, a manual inspection was performed by the researchers, mostly medicinal chemists, to select 100 compounds for the experimental validation. The validation confirmed that 8 compounds were found to modulate the beta-2 adrenergic receptor activity. This highlights a validated workflow using different modules of SILC's small molecule suite that can be used in a structure-based drug design campaign. Okay, now let's dig into the new updates coming in version 2024. The major updates in version 2024 include GPU acceleration in Monte Carlo docking program, upgrades to the GUI for more intuitive experience, such as addition of CGNFF suite in the GUI. A lot of additions in this command line interface, some of which we will talk in detail next.
Let's take a detailed look at the SILCS-MC upgrade. Since 2022, we have already moved the GCMC and the MD simulations to GPU. In version 2024, we transferred SILCS-MC to GPU. Now we can finally perform both SILCS GCMC MD simulation and the SILCS-MC on GPU. We have tested 1,000 ligands with the average size of 42 addons with both CPU and the GPU versions of SILCS-MC for the performance comparison. The CPU version of SILCS-MC was performed on a single-core CPU, and the GPU version of SILCS-MC was performed with NVIDIA video card. For SILCS-MC post-refinement, the GPU version decreases the running time from 8.5 seconds per ligand to 4.0 seconds per ligand, which decreases the computing time by 53.5%. For SILCS-MC docking, the GPU version decreases the running time from 118 minutes per ligand to 35.2 seconds per ligand, which reduces the computing time by 99.5%. In the normal procedure of CPU version, the docking job split to 5 parallel runs on 5 cores, yet the job still takes about 23.5 minutes per ligand. The GPU version has a significant improvement on the performance in terms of speed and the amount of post-sampling. We also did the same comparison on AWS. Both the CPU and the GPU instances shown here are common nodes used by many customers. For post-refinements, it takes 1 hour and 17 minutes to finish 1,000 ligands on the CPU instance, with the total cost being 43 cents, and it takes 2 hours and 15 minutes to finish on the GPU instance, with the total cost being $6.85. For docking, it takes 163 hours and 7 minutes on the CPU instance, and it takes 19 hours and 51 minutes on the GPU instance. Although the total cost of both are almost the same, the GPU version is about 700% faster than the CPU version. For a regular protein with the 400 amino acids in the system of 75,000 atoms, it takes 36 hours on 10 of the P3 instances with a cost of $1,000 in total to generate the SILCS frag maps. Indeed, the simulation cost is expensive. However, with a set of frag maps, users can do SILCS MC post-refinement and docking at a very low cost. With the same 10 P3 instances, it only takes 15 minutes and $7. And the more ligands you perform, the average computing cost and the time per compound drops significantly. Again, the SILCS methodology produces free energies with FEP level accuracy with more than 1,000-fold speed increase for ligand ranking ability. It only takes minutes for each ligand in SILCS MC versus about 1 day for FEP. Combined with Bayesian machine learning, as in the Go paper published in 2021, the SILCS methodology can achieve similar free energy results as state-of-the-art FEP tools.
SILCS BML, where BML stands for Bayesian machine learning, is our next major addition that allows leveraging experimental data to improve ligand ranking. Now, we can run either SILCS MC docking or pose refinement calculation to generate ligand poses and LGFE scores. At this point, if we have any experimental data for all or some of the ligands, then it can be used to perform Bayesian machine learning optimization of the frag maps. This is typically helpful in case of ligands in a congeneric series, as the GFEs assigned to each atom in the set of ligands is getting reweighted to give you better predictions. The next molecule you design in that series will be ranked with much higher confidence. Next major feature we have is addition of command line interface support for performing the pharmacophore screening. As described earlier, users can directly transform the SILCS frag maps in a binding site to target-based pharmacophore features using the SILCS farm module, and save the pharmacophore features as PH4 files. Now, we also support running the pharmacophore screening on large databases for these hypotheses using the open source program Farmer integrated in the SILCS farm module. Farmer needs pre-prepared databases to perform the screening. Users can get ready to screen Farmer compatible databases like Inamin Real, Molport, and Cambridge from us, or users can build their own database for which SILCS Bio team can assist. With a single command, as shown here, users can perform the pharmacophore screening. For a database like Molport, this takes about 20 minutes on a single core CPU, while the process can be parallelized on multiple cores to reduce the screening time further.
Now let's discuss our new best protein ligand complex retrieval tool. In traditional docking and FEP calculations, the protein is present. However, in the SILCS workflow, LGFE calculations and SILCS MC docking are determined based on the overlap of ligand atoms with frag maps rather than based on a rigid protein. So when we view the docked ligands overlaid with the static input protein, we might not get the best fit with respect to the initial protein structure. With our new protein ligand complex retrieval tool, you can extract complementary conformations of the target protein with your docked ligand and analyze key protein-ligand interactions. To do this, all you need to do is add one extra flag while generating frag maps to collate the protein trajectory from the SILCS simulations, and then run one simple command to scan this protein trajectory for conformations that best complement the docked ligand structure. After that, you'll get the most energetically favored 3D structures of the protein ligand complex and a CSV file containing information about which frame the protein conformation originated from, and the electrostatic, LJ, and total interaction energy of the protein and the ligand. Looking closer at the most energetically favored structure from our example case, we can see how well the docked ligand fits within the extracted protein structure. This structure could be analyzed as is, or we can perform MD simulations to extract more data with our new classical MD simulation module. So what can we do with our new classical MD simulation module? Well, now you can set up, run, and analyze MD simulations for proteins, protein ligand complexes, and covalently bonded protein ligand complexes in solution with neutralizing ions or any salt concentration you desire. And you can also do this for membrane proteins as well. You can set up, run, collect, and analyze your MD simulations with just 4 simple commands. The simulations will include energetic minimization of the initial system, a 3-step equilibration MD, and a production MD. And upon completion of the MD simulations, the 3 analyze command will perform an array of standard MD analyses, including RMSD, RMSF, radius of gyration, SASA, and if you have a protein ligand complex, protein ligand interaction energies. Finally, also making its debut to the SILCS Bio platform is MC docking along a path. Using the SILCS MC slab mode, you can now dock your ligand along a linear path of your choosing. From this, you can get the most energetically favorable conformations of your ligand along a path, which can be used to identify potential target binding sites to block gateways to active sites. Or you can also extract LGFEs along the pathway to characterize binding and unbinding pathways. And those are the additions to the SILCS universe. Please sign up for our newsletter to hear about our future additions to the SILCS universe, which will include SILCS Covalent for irreversible inhibitor optimization targeting cysteines, SILCS hERG blockade for rapid prediction of ligand binding to hERG channels, SILCS permeability for rapid prediction of ligand passive permeability, SILCS P-charge for protein effective charge prediction in different salt environments, and SILCS water for docking ligands with waters explicitly accounted for. Overall, our future goals are focused on combining the SILCS physics-based frag maps with AI technologies to streamline the drug design and discovery process. Thank you for tuning in. We're excited to bring you version 2024 of SILCS Bio with improved performance and new functionalities. For more details on our new features, please visit docs.silcsbio.com. And for general inquiries, please visit us at www.silcsbio.com. We'll be happy to take any questions.
Great. Thanks to our technical team for putting that together. So yeah, like Oscar said, we'll go into questions next. Let me start filtering these out. So first question is, does SILCS currently support or have plans to incorporate specialized identification and annotation features for diverse classes of proteins, such as antibodies and other functional protein classes?
Repeat that question?
Yes.
Yeah. Does SILCS currently support or have plans to incorporate specialized identification and annotation features for diverse classes of proteins, such as antibodies and other functional protein classes?
No, the software can handle a variety of classes of proteins, say globular proteins, membrane proteins such as GPCRs and so on, proteins with common metals bound and these types of things. We have no plans to sort of set up some sort of a classification scheme, but the software is usable on most classes of proteins. Even on ion transporters and these types of things, people are using it now.
And next question is it possible for customers to access the new version using their existing license? So I can answer that. We'll have to send a new downloadable link for you to access this newest version. We'll reach out more tomorrow though for we need basically some information to make that downloadable link for you. So reach out tomorrow, but no, using the existing license, you can't. Okay, and then next question is, can we use molecules with metal atoms for docking?
At this time,
Yes. I mean, certain, yeah. If your molecule is linked and CGNFF can process it, then we will be able to talk. But yeah, not like a coordinated metal ions, I guess.
Next question is, I'm interested in protein charge. Is there a way I could get that early?
Yeah, so for protein charge, you can use the SILCS P charge that is currently in development. You can contact us at, I think it was info@silcsbio.com, and we can give you a beta version to test.
Next question is, are there any resources you can recommend for me to get an understanding of how to use these features and these new features? So I can take this one. So yeah, like they were saying, you can see here docs.silcsbio.com that basically goes over the workflows for our new features. And then additionally, we can also just set up one-on-one calls with you and go over specific workflows that you would be interested in learning more about. So if you'd like to do that, just feel free to reach out to me. And the next question is, we sometimes find dozens of hotspots for brand new proteins where we don't know any of the ligands. How do we prioritize the hotspots for virtual screening?
I can take that. Right now, we typically will look at the hotspots. We'll identify those hotspots which have adjacent hotspots that are within approximately 10 to 12 angstroms, and we'll prioritize those. We can then go in and do SILCS MC on a set of about 350 FDA-approved drug-like molecules. And then from that, we get average LGFE scores for, say, the top 20, along with their buried relative solvent accessibility, and use that to rank sites. We're also currently developing a new method to more automatically identify drug-like sites based on the SILCS hotspots using a combination of machine learning along with the hotspots themselves. And that will greatly facilitate identifying new sites on a protein to consider for virtual screening.
Next question is, I'm a SILCS Biologics user. Were there any improvements in the SILCS Biologics capabilities?
So not directly with SILCS Biologics, but since we improved the SILCS MC docking with the GPU acceleration, now your hotspots calculations incorporated in SILCS Biologics will be faster. And since we also have more pose sampling, you'll also have more accurate excipient poses too.
Next question is, does the ligand protein complex retrieval always find a reasonable protein conformation for a given ligand pose? Or is it possible that some ligands would not fit in a given binding site?
Well, it will always find an approximately acceptable conformation, although there's often still some steric overlap. Thus, we have a procedure which does a restrained minimization to allow for the protein to relax around the ligand conformation. So the ligands are suitable for that site based on the SILCS frag map. It's just finding an exact conformation that minimizes the steric overlap and maximizes the interactions. But there could be still some steric overlap, and so we do a minimization equilibration protocol to account for that.
Yes, and we have like a way to do it, so since we have 100K frames that we need to scan, you can skip certain frames to get results faster, but also you have option to look at the best top 5 or top 10 poses. So you have option to look at where exactly the side chains of protein are preferred in that pose of ligand over certain frames. So there are multiple ways you can kind of analyze those last top 5 or top 10 frames that you get.
Next question is, for the slab mode in SILCS MC, letting users define their own paths, do you see a decrease in accuracy through user errors in defining the path? Oliver, you want to take that one?
Yeah, that's a good question. I mean, the accuracy basically is the same as regular SILCS MC docking. The differences in the SILCS MC slab mode are user has to define the starting point and the ending point. Also, the sampling range is in many cylinder slabs instead of just one sphere. And also the LGFE fluctuation, like we shown in the chart before, reflects the more favorable and the less favorable sides through the entire binding channel.
Next question is, can the new version of SILCS Bio build different types of lipids and different percentages of lipids/cholesterol for a membrane protein in the latest classical MD module?
The answer for that is no. However, if you'd like a different type of membrane, please contact us at info@silcsbio.com and we'll work on that with you.
And we kind of answered this a little bit, but maybe we can go more in depth. So I'm curious about the Bayesian machine learning module. What's the model setup like? What kind of data does it need? And how does it improve the accuracy for predicting ligand binding?
Yeah, I can take that. So yeah, as we showed in the slide before, the experimental data that you need is something like delta G values or IC50 values, which can be converted into a binding affinity like data. And then basically it uses that information to do a better ranking of your ligand affinities, right? So what we are doing at this point, we have different targets that we can optimize with. We can target the percent correct values. We can target the Pearson correlation or the RMST between your experimental data and the LGFEs. So if you go on the documentation site, you'll have more details about how you can decide on that. And at this point, we are basically reweighting refining the weights assigned to each ligand atoms for that particular frag map type for that congeneric series, right? And that gives a better prediction along that congeneric series. And then when you do an additional ligand optimization to R site, you already have a data that is well frag maps that are basically giving you much better prediction. So that's kind of the framework underneath that happens.
But okay, the SILCS frag maps are often giving pretty good predictions already. So you might be up in like an R of 0.6, 0.7. The BML optimization will often maybe improve that like by 0.05, 0.1, like from 0.6 to 0.7, and so on. So, you're starting from something which is working well. So you do make improvements, although oftentimes the improvements are not, huge.
Yeah. Yeah. See, in this case, the R value has upgraded from 0.57 to 0.66, while the percent correct already was about 0.73. That got improved to 0.75. So depending on your experimental data range, how diverse it is in terms of the range, and based on that, it can help your BML optimization better.
Next question is, just wondering how user-friendly the new GUI updates are. Could someone with limited computational chemistry experience, like some of our medicinal chemists, use it? You want to take that over?
Our SILCS biographical user interface is actually pretty user-friendly and intuitive. So basically, with a GPU setup server as a backend, you can submit the jobs and collect the data and analyze the results in our GUI. You can also visit the documentation website at docs.silcsbio.com to follow the instructions for all the tools we have, or you can contact us for a demo or video call.
Yeah, typically like one session of like going through doing modifications and doing the calculations will allow a person to get up to speed very quickly and then be able to do their own calculations going forward. It's very user-friendly.
Next question is, does SILCS Bio have any plans to implement more AI and machine learning in the software?
In general, what we've been doing is sort of doing a combination of the physics-based SILCS frag maps with AI. The one example is the Bayesian machine learning type of an approach. We we've published a paper where we've used the SILCS ligand grid free energies for molecules traversing lipid bilayers. And we've combined that with an AI model to rapidly predict drug permeability. We have a covalent model where we use the SILCS information combined with an AI model to select ligands, functional groups to target different warheads. We also have a model where we combined the SILCS frag maps on the hERG protein, and this is a drug liability model to better predict binding to hERG. And so we're now sort of developing a number of areas where we sort of combine the SILCS frag maps, the physics-based information with AI to sort of jump the accuracy of the method up, increase its applicability and so on. And we think that's going to be a good path going forward as we continue to develop the SILCS technology.
Next question is, so new to SILCS here, can you explain more what that frag map you mentioned gives?
Good, Abhishek?
Yeah, so as we mentioned, the frag map is basically a pre-computed ensemble which is representing affinity maps. So we have a whole lot of probe molecules in the simulation that are interacting with protein and trying to occupy those very favorable sites. So in the end, we look at all the probabilities of different voxels and find out how each probe how much each probe has been able to sample that particular voxel in and around the protein, right? And based on that information, we can basically calculate a grid-based free energy, which in the end, the files of map files basically look like electron density maps, which have all the information about different functional group types at each of the grid around the protein. So the frag map is going to give you whole qualitative understanding as well as downstream, you can use it for quantitative understanding of that target that you're working on.
Next question is, for best pose retrieval function, is ligand force field converted to CGNFF to run follow-up all-atom MD simulations? Is there integration with FF param?
Yes, I mean, the ligands are always in CGNFF format. So yes, you basically have a ligand in CGNFF format. You combine it with the Charm 36M force field, and then you set it up to run your MD simulations and so on. At this point, there's no direct integration with FF param. I mean, FF param is integrated with CGNFF, so that's already there. FF param just works in the small molecules. It doesn't work with protein. So it's all focused on the small molecule optimization.
Maybe we want to go more into depth to this question. So the upcoming SILCS water functionality mentioned that MC sampling will happen with explicit water. Is this referring to the step where you sample the ligand conformations in the field of the LGFE maps?
Yes, what we're doing with SILCS water is we can we'll know the binding site, and we go in and we take the ligand and we take this is ultimately user selected, but the default approach is then to include 15 water molecules. You then do your exhaustive SILCS MC docking of the ligand and the water simultaneously. From that, you get configurations of the ligand with the water. At that point, you go in, we have criteria to select a subset of the waters based on energetics and distance criteria. And then from that subset of waters along with the ligand, we can estimate the ligand, the overall binding affinity. We also can map the distribution of the waters around the ligand. And what's particularly interesting is that we also have the GFE scores. So now we have the GFE scores for the ligand, which is telling us which atoms in the ligand are contributing to the binding affinity. We also have the GFE scores for the water molecules. And it's interesting that you'll see some of the waters that have like a favorable interaction with the ligand, they may actually have unfavorable GFE scores. And that indicates the water molecules that if you add a functional group to the ligand to displace that water molecule, that's going to give you a favorable energy gain. Now, there's other methods out there that identify similar waters, but with the SILCS water methodology, we're actually identifying those waters in the presence of the ligands itself. So this is very powerful and clearly represents an advance that's not accessible to other computational technologies. And again, the SILCS MC with the water molecules, it's about 4 times slower than SILCS for the ligand alone, which is, again, on the order of minutes. So you can do many ligands with explicit water and so on.
Next question is, I've mainly been using SILCS for new targets, but was wondering, can you explain how I can use it for existing targets as well?
Well, I mean,
Yeah.
the advantage of the technology is that you can go in and you can try new ligands at an incredibly high rate and an accuracy, similar to that of full-blown FEP. So the SILCS GUI has methods that allow you to, tools that allow you to go in, visualize your ligand in the presence of the frag maps, to select regions of the ligand to do modifications. The software is set up to do hundreds of modifications, and then you can submit the jobs, and you'll quickly get the energetic information back that you can use to drive your ligand design. If you want to use some other software tool to generate ligand modifications, new ligands, and so on, you can do that. You can then use the SILCS MC to run those ligands, which can be in like MOL2, SDF format, and so on. So there's a number of ways that you can use the SILCS capability to move a project ahead in the optimization stage, Or you may be at the point where, okay, we've been focusing on one site. We can use our frag maps. We can just use those same set of frag maps to go back. Maybe there's some alternate allosteric sites we haven't considered. That information is already there. You can quickly run the hotspots, do that type of analysis, and so on. So once you've got your maps, you're sort of like good to go with a number of uses to help you move your drug discovery process ahead. And again, it's rapid. It's at an accuracy level of FEP. In addition, you get that GFE information for individual atoms. So when you modify a ligand and you add a functional group, you might see that the functional group gains you some energy, but at the other end of the molecule, it might lead to losing some of the binding free energy. You can actually visualize that using these GFE scores. So the information content of the from the SILCS calculations is much higher than that of the like FEP and other types of methods, because you're understanding the actual atomic contributions of the different atoms in the system and how they balance each other as you're doing your ligand design.
Yeah, I think we did not mention one additional thing about pose evaluation. So I know a lot of our current users also use other tools, and sometimes mostly based on the question, like people run silk simulation, use them for qualitative analysis of the target, but then typically don't do a whole lot of downstream processing that silks can do efficiently, right? So in case like you come up with a pose somewhere else and then just want to evaluate that pose, we now have a pose evaluation mode in the SILCS MC calculation that you can use to get that atom-based grid-free energy just for downstream analysis. So that's a good addition, I think, in case someone is interested.
Looks like we have time for one more question. So this one says, what sort of service work do you do? I was only familiar with your software products. So I guess can we just elaborate on service work we've done?
We do a variety of service type of work, in the context of the SILCS technology as well as molecular dynamic simulation. So we can go in, if you have a target that you're interested in, but you don't have the local capabilities to do the SILCS simulations, we can do that. We can then follow up and do all the hotspots analysis, identify new binding sites. If you have known ligands that you've been working with, we can evaluate them for their relative binding affinities. We can identify new sites that they bind to. We can also do service work in the context of the SILCS biologics technology and so on. If you're interested in just like MD simulation type of work, which isn't necessarily using the SILCS technology, we can work in that area and so on. So we're quite flexible with the capabilities that we can supply in the service area.
One of the recent service projects we did was based on a glycoprotein. So those are very difficult to model, and there are not a lot of tools available to build glycoproteins for MD simulation as well. So we can do that as a service project if someone is interested. So just send us an email at info@silcsbio.com.
Yeah, so we still have a couple questions left, but we don't have enough time. So I'll email those individuals separately, the answers to those questions. But if you have any questions or feedback about this webinar, about the new updates, feel free to email me. I put my email in the chat. We can schedule a time to talk and kind of go more in depth over them. But thanks to everyone for joining. Yeah, enjoy the new SILCS release.
Thank you.